Okay, sir, I think we can go live.
Right.
We are live? Okay. So, welcome to another episode of SciComm Synapse, where we bring inspiring stories from the world of science research and innovation to young minds aspiring to build careers in STEM. Today, we are honored to have with us here Professor SC Lakhotia, a distinguished geneticist and an eminent scientist from Banaras Hindu University, whose journey represents more than 5 decades of curiosity-driven research, mentorship, contribution to Indian science. Professor Lakhotia has made pioneering contributions to field of gene regulation, chromosome biology, and RNA biology, particularly through his extensive research on long non-coding RNAs in Drosophila. His work has helped us shape our understanding of how non-coding RNAs influence cellular processes, stress responses, and development. Beyond his scientific discoveries, Professor Lakhotia has played a remarkable role as an educator and mentor, guiding generations of researchers and contributing to conversations around research ethics, science policy, and the future of scientific education. In today's conversation, we'll explore his journey as a scientist, the importance of pursuing unanswered questions, the challenges and rewards of academic research, and his advice for young students who dream of becoming the scientists of tomorrow. So, Professor Lakhotia, it is a privilege to have you with us. Welcome to SciComm Synapse.
Thank you. I'm happy to be here. Yes, it's a pleasure.
Okay, so I won't take much time and will like to start with questions. So we hope this session to be really interactive. So audience, you can also send your questions to the in the chat box, and we can pick it up at the end of the podcast to ask the questions you need to ask. So, so your career has spanned more than 5 decades. Looking back, what was the single decision that changed the trajectory of your scientific journey?
Well, actually, my research journey started 6 decades back in 1966, more or less. But then, to get into this field was something I'm happy to share the embarrassing student that I was in my school and the family. But then I wanted to be a medical researcher from my childhood. I won't go in detail why, but that was my ambition, that to be a medical researcher. And so I took biology in my final years of school education in Calcutta. But then the results were devastating. I had a poor 54% marks in the board examination, and having no money, neither merit, I opted to take up BSc studies. And I think that was the turning point. That's when I decided that I'll be a teacher and a researcher. And I have no regrets. I'm really indeed happy that that thing happened, because that gave me a challenge to prove myself and to enjoy what I've been doing. And that's the 60 case, my research journey, which BSc started in 1961, then MSc from Calcutta in 1965, 66, and then joined for PhD in Calcutta with one of the best young teacher researcher who actually shaped my life in many, many ways. As curiosity-driven research, independent research, and the freedom to ask questions and go ahead. So that's how I think the beginning of my my research journey and teaching journey started. And you must understand, as of today, what AI generates is what information humans have generated, have put in the internet, and which is available as software, digital material, and they can be humans of all kinds. Some do good research, solid research, others do, and we know that number is increasing today, fake research. And that fake research also get collected by AI, and that's get transmitted to us, and this can be very dangerous. So I would say that be dependent on AI, make use of it, but then very judiciously. Find out what the information that will turn while, and that's where one needs good understanding of basics. And of course, language. I mean, you see, two things that are missing in today's young generation is language. Thanks to all the social media concepts, we don't speak, we speak either monosyllables or in what you call smileys. But we need to learn, we need to communicate, and that's where we need good language. Not necessarily grammatically correct, but at least we should know how to express what we are thinking, what we are understanding, what we are wanting to communicate. And that's where a good language is necessary. I won't believe on chat GPT or something like that in improving my language. No, because then it becomes a monotonous language that everybody uses. No individuality. Every individual has to have, like sometimes when I read a paper without knowing the author, I can make out who the author may have been. But each author has a particular style, particular pattern of expression. So I think that's what I would suggest, that don't be dependent on AI. Have your own questions, look widely, but before getting those questions, read more deeply. Read and read and read. That's what my advice will be. Or if I were to start, that's how I would want to start all over again today. I still keep on reading enormously because I still do not know many things. And one thing that I very commonly say is, when you know something, what you know is what you still do not know. That's a motto to be kept.
So, sir, you pointed out a really good thing that in the world of AI today, we are losing out the essence of expression that each scientific author might have. That's a very nice point to be taken from this. So, sir, as you said, talked about the current generation, how they are talking in monosyllables. One more thing which is variedly seen in, I won't say in our generation is that, so there is this fanciful thing of going out of India. So they are worrying and they're falling under the pressure that do we need to go and study in abroad to do an impactful science, or is it fine we are doing science from India? So from your experience, I would like to know, building a world class research program in India, what advice would you give them?
Well, yes, you see, I mean, I can tell you when I started my PhD or finished my PhD, there was pressure on me also that I should go abroad to do postdoc. I was not very keen. The reason being, I had believed that yes, in India also we can do quality research. We can do it, but yes, it's difficult because partly the way our infrastructure has been developed, the way our total social milieu has been developed, we sometimes still think that if your passport has stamps of many countries, you are better off, better intellectual, which I don't agree with. Yes, you do get more experience, you get wider knowledge, or had gone the postdoc. Yeah, I did also go for 1 year, but that was after I had my job. So I got my job very soon after doing PhD, after 1 year of postdoc in India, and then got a job. But then yes, it has been a struggle developing things. But once you keep on struggling, you build up a ecosystem with like-minded people, develop collaboration, develop interactions. Yes, I think we can and we must do world-class science in India itself. If you want really India to be so-called real developed country. Otherwise, we'll continue to be what I say. We also ran, but we never get to the front rank. And that can happen only if we begin doing things with confidence within the country. And I think that's doable, that's feasible.
So, sir, you began studying non-coding RNAs very long before it was the hottest topics in biology. How do you stay confident in pursuing questions that many people initially overlooked?
Okay, well, you see, this is more of a philosophical as well as intellectual question, partly because I did study biology as biology, zoology, botany, and physiology, in a classical way. So I was aware, I had belief in evolution, I had belief in biological diversity, and I knew that all biological systems do not achieve the same end following same path. They can achieve the same end by using very different pathways. That's what I learned when I was doing a PhD on doses compensation, that is X chromosome activity equal in male and female. Mammals do it very differently, and we discovered Drosophila does it very differently. End result is same, that 2X versus 1X chromosome, female versus male, the expression of those genes is equal. But then, so I knew that biological system need not follow a monotonous path. But the first, my confidence about the so-called non-coding RNA, that term came much later, was when I found in 1973 that the classical heterochromatin of Drosophila is transcriptionally active. It was not believed by anyone. In fact, one of the reviewers said this is an artifact of electron microscopy. And my answer was, I was confirmed my experiments. My answer was that if it is an artifact of electron microscopy, then EM should not be used in biology. It was accepted. And that observation stands till date that yes, heterochromatin can be transcriptionally active. And then in 82, when we discovered that the gene that I was working on, that I got interested in, was not making a protein, it was disappointing in a way, because then, and you see more disappointing because this was in 82, and in 1980, Crick and others, top leaders in molecular biology, had started selfish DNA, junk DNA, something that doesn't make protein, it's selfish and junk. But then I never believed in that concept. I continued, and I'm glad that in India, I could get some funding for continuing to work on this. My competitors in MIT and Heidelberg, they didn't get funding working on a gene, on the same gene that I was working on. They didn't get funding because it's non-coding and supposedly selfish. But I'm glad we did it. And so way back in 1996, I had predicted that non-coding RNA is going to be very, very critical in coming 10 years. And I'm happy that that happened, yes. So the point is, believe in what you are seeing, believe in what you are observing, and keep, do not just follow what is dogmatically suggested by everyone. Because if you find something that fits in with existing knowledge, we don't make an advancement. But if you make an observation that doesn't agree with existing interpretation, that's where you make a new discovery. And that's what I think has to be the motto of science.
So, sir, you talked about disappointments. So, one more thing, as in research is full of failures. We have failed experiments, like more than the successful ones we are facing on daily basis, the failed experiments. So, can you share one experiment or project that you planned it as in a way, but the outcome was very different? And what was the like outcome? What it taught you?
Okay, well, I think I'll go back to this non-coding RNA gene that I was working on. You see, my observation on this gene started when I was doing my PhD. It was a serendipitous observation I made that when I used benzamide, then a very known inhibitor chromosome transcription, that inhibited transcription chromosomes all over in Drosophila cells, but then activated one single gene. This was way back in 1969. And that this was not my part of my thesis work, but it caught my attention. And I thought I should follow it up because at that time, see, 1969, we knew the genes make RNA and they should make a protein. But then a direct gene to protein, that this gene makes this protein, was not known. And what kind of RNA it makes was not known. So I thought this was a beautiful model system that I have, where I can activate just one gene, and then I can follow to what the protein it makes and how it makes and so on. And I thought when I take my independent position, I'll start working on this. And so that was supposed. But then initially 1 year I was at Bardwan, couldn't develop much lab facet. Moved to Gujarat University, and then immediately went for 1 year to Edinburgh. That's where I did my EM studies, heterochromatin transcription. Came back, and I thought, okay, now it's a simple experiment that we label the tissue with radioactive amino acids, separate the proteins, and see and do an autotography and see which protein is newly made in response to this. But the problem was we didn't have money to buy a gel electrophoresis system. We didn't have money to buy a gel dryer. And we struggled and struggled and struggled. Tried all kinds of things, how to make it. Finally, in 76, I moved to Banaras, kept on trying. In 1979-80, one student from Calcutta joined my lab, Dr. Mukhopadhyay, and he took upon himself to assemble a slab gel electrophoresis system, to assemble a gel dryer. We had no money to buy those things commercially, but he assembled. And then finally in 81, late, when we got the first result, the disappointment, you don't see a new protein. Okay, you have the gene active, but you don't see a new protein. And that's where the idea came, that's non-coding. And I think all these years of kind of frustration became a beautiful day. And you know, these things happen, but what you need is, I would say, perseverance and of course resources. But if you don't have resources, build your own, and that's what we have been doing. That we make our own many things, improvise things, and things work.
So, sir, you have mentored, as you said, like there have been dozens of PhD students that you have mentored. So, what are the qualities that separate students who merely complete their PhD from those who become independent scientists as
Okay,
or researchers?
uh, that's a little difficult question, but then I'll try to say that because it's not very easy to make clear chromatographic separation that okay, this is good and this is not good. Each one will have some variations and some individual strengths and spiritual weakness. But in general, we must understand PhD to me is a training period. Training to learn to ask questions, define questions, build hypotheses, do studies, analysis, experiments, or theoretical to check whether a hypothesis is correct or in its modifications, and then make an interpretation in context of what is already known and how your development happens. So that's what it is. Some students come to get a degree. They need a degree. I don't say that those who are even curious about science don't need a degree. They also need a degree. But for them, degree and knowledge both have to go together. But for many others, they just want a degree. And unfortunately, that proportion is increasing in current times because of various reasons. But then when you look at the student and see, okay, the student is confident, what I personally prefer is with my PhD student, I, I normally do not say do this experiment and do this experiment and do this experiment. I would simply put my idea that these are the questions that we want to ask. And then the student has to develop the ways we keep on discussing, we keep on arguing, we keep on learning that, okay, this may work, this may not work. But then that's how those who have gone through this process, I'm very happy that many of them have risen to very top positions in their own careers. So that is what I think. A simple PhD versus the PhD which is really earned by learning to ask questions, learning to define how to answer those questions, and then interpreting them. It's they who can will make a difference.
So, sir, in today's competitive world, many students also feel the pressure to chase publications, the impact factors, and CV building. So, sir, can you, in your language, redefine success in research for young scientists? What would it look like? Or is it exactly what are we feeling like that we need to go for impact factors and journals and publications, more and more of publications?
Yes, unfortunately, the current times have become so loaded with this kind of quantitative metrics that the pleasure of research is gone. Everybody is running to meet the target, and the target becomes the objective, not the objective to get something new. And the result is we have all kinds of unethical practices happening, paper mill journals and the predatory journals, fake authorship, purchase authorship, sold authorship, all kinds of things are happening. Why? Because I agree that current ways of assessing a young person or even a senior person is quantity. How many papers published? What impact factor? To me, these are wrong parameters, completely wrong parameters. I know I'm completely wrong in terms of people who are now doing the assessment. I've been fighting them as well that the way they are doing this is completely wrong. We need to measure the quality of impact of the research, not just the impact factor. Because we must understand, impact factor is an arbitrary value which can be easily gained, which can be easily manipulated, and most journals are now manipulated. Authors are manipulated. You see, I decide with my friend, I cite you and you cite me. Our citation goes up, and the journal editor says, cite papers from this journal, and therefore the journal impact factor goes up. I mean, because I've been working on ethics also quite a bit, writing about it, analyzing it, I know what all games happen. And that's what I would want to caution the young generation that yes, you will have problems, but then if you stand your ground, if you are good in your quality and confident about your quality, you will find a way. But do not get swayed by these quantitative numbers, because they are really not telling you. I mean, I know there are authors who have several hundred papers every year. I don't know how one can do that. There are PhD students who, by the time they finish PhD, have 30 or 40 papers. I don't think that can happen in a qualitative manner. So I would be happy even if I have 3 papers from my PhD, but they must be good papers. I mean, like my own PhD papers, I'm still proud of my first research paper, because that had a quality, and in my sense, and not just in my sense, it may not have high impact factor, but it did have an impact on the thinking, on the process at that time and even today. So I think long-lasting pleasure, long-lasting respect comes from quality, not just quantity. So the young generation, I know they are facing a very difficult task, because they are sitting on the other side, and we who are sitting on this side are doing wrong things. But that's where the young generation to protest, find a way, and if they combine together and they can think in that sense, I'm sure things will look up better, and that pressure will disappear, a wrong pressure that is happening. Because, as I said in the beginning, that kills the pleasure of doing science. We have the pleasure of knowing something unknown, but now you are not worried on that. You are worried on how can I have one more paper, and that's not the best thing to happen.
So, sir, as in, what excites you now? You have done so many things. So, what are the next thing that you excites about the future of biology? If a student starts their PhD today, which big scientific questions do you think that will define their journey of next 20 years, 20-25 years?
Well, biology, I think, is continued to be the most exciting for ever since people started experimenting in biology. See, from descriptive, we have now become to experimental. And thanks to the remarkable developments in physics, chemistry, maths, and technology, and the kind of system that we have available now, the kind of question that we can ask, the facility that we have, it is becoming one of the most exciting domain of life. To say that this area is better than others, I won't venture to do that, because any question in biology, if you're asking the original question, can be very exciting. See, one simple thing that what you are missing, for example, in India is there's so much of biodiversity. We're not looking at that. We need to know, everybody is trying to become molecular biologists. Yes, be a molecular biologist, but don't forget that you're a biologist. Biology is not just molecules. Molecules are only part of biological system. And this is where I think we are neither teaching them typical organism biology, and because we don't teach them organism biology, they never look at the cell or molecule that the young person or the even senior persons are looking at in context of biological system. I think that's where the important point comes. Certain areas are certainly most exciting: neurobiology, the understanding brain, understanding any of biological system, how to maintain our coordination, how to maintain how the body maintains what it is, no matter what happens, the homeostasis. Now, all these are really remarkable questions. How do organs live in deep ocean under such a heavy gravity pressure, very high temperature, very low temperature? All the more I look at it, I actually enjoy watching videos about diversity of life at various places around the world, and I feel I wish I were 20 years old today to start looking at these questions immediately. The question, but that's what one has to appreciate the diversity, and then begin to ask question. Not to say, okay, these are fashionable things. There can be certain things that will have immediate application. For example, from a medical point of view, that's one part. But then, as a basic biology, no matter what we claim that we have learned so much about biology that we know how development happens, which genes become active at what time, and so on. But believe me, we are really at tip of iceberg. The life is much more complicated, much more diverse, much more deeper things to analyze, and that's and any person who has curiosity to ask these questions will have remarkable ways today to answer those questions. And anybody should be excited to look in biological issues.
So, having said that, sir, you're really good at mentoring and everything. So, imagine you're speaking directly to a first year college student who dreams of becoming a professor one day, but is not very much of good in study, is an average, confused, and overwhelmed, but is really curious to study science. So, what would you tell them? How would you like motivate them to go ahead and pursue their dreams of becoming a good scientist or researcher?
Well, you see, the question of mediocre is a relative term. Somebody may be mediocre in something, but may be very good in something else. So, what an important point, like you see, as I said at the beginning, my own career as a less than a mediocre student, and right, and in a family, big family, where all my elders were bright, topper students. So, but then that challenged me. That did and I had my own conviction that yes, I can do it. I have to. So that's one part. See, to be mediocre, there can be multiple factors for this. But then what one needs to define is where your heart lies. See, if you think I'm a great scientist, all right, fine, let's accept that. But you ask me, okay, being very bright, write a poetry. I'm absolutely useless there. Okay, as an artist, as a painter, I may do some kind of, but then so each one has one's own capacity to do something, and that's what young person and their parents and family need to identify. So what's happening wrong is parents and social structure believe that bright students must be either engineers or doctors. Completely wrong, absolutely wrong, because this is killing the individuality, the capacity of the young person, and the social structure. The result is, for example, now what we see for few thousand seats, we have lakhs and lakhs of students appearing in tests. Obviously, all of them cannot qualify, and because parents put pressure, their family, the others, relations, teachers put pressure. Many of them succumb and lose their life. Now this thing must change. We need to identify what one can do. Everybody need not be a researcher. Everybody need not be a teacher. Even those who have got into, let's say, basic science, they can in multiple ways, and they need to identify that you see somebody may be a good writer, good communicator, not a good researcher. So that's what one has to identify within oneself. What interests me most? Where is my best capability? There are, we know many examples. People did like I know one student who was topper in our class at masters, but then said he's going to go into banking, and he's a good banker today. I mean, and somebody went for writing. They're good communicators. So it's not that because you have done your PhD, therefore you are bonded to one path. No, you can always diversify. You can always choose what you want to do, but the important point to do is you have to do that with conviction, not under compulsion. See, research or any job needs passion, not compulsion. So you have to be passionate about what you're doing. And so if a young student, if they were to learn to become scientists, let's say to become scientists, I would say first thing they need is curiosity. Do they have are they curious? Are there something burning inside them that okay, I want to know this? That's the most important thing. Then they must have desire to learn, desire to read, and they need to develop a much wider basic learning than what's happening today in classroom. Unfortunately, classrooms are not teaching into basics. I know what's happening is you read one paper and you think you read the subject. You have forgotten that there are something called textbooks. You see, textbook build up things from base to and we now don't know. I mean, I know in my own lab, I have hundreds of textbooks, but they're hardly read by students. And keep on telling them that look, this is where real knowledge lies. Maybe it has become older, but you at least get to know the base on which you build. So the important point to do is curiosity, desire to learn, and learn deep. See, any knowledge has to be pyramidal. We have broad base, then we focus on the question that we want to ask. But if you don't have broad base, then the question becomes superficial, and that's the important point. But then what you need is also confidence that yes, you want to do it and you will do it. There will be difficulties. Say no career is smooth going as such. Every career has its own what we call as occupational hazards. So we must be conscious of that. And of course, research, as you said, failures are part of research. We learn, we think, we design a bright experiment, and it turns out, okay, it doesn't work that way, that way we hypothesize. So the bright experiment fails. So these are there. And that's where we need perseverance. But okay, failure shouldn't frustrate us. That's where we learn. And of course, hard work. We can't just do 10 to 5 research. And of course, the other thing that I would say is spend less of your energy on your so-called smartphone. That makes you unsmart, which I know is something that young generation thinks it's a completely wrong part, because that's where their life seems to concentrate. If they don't have, I mean, actually, why should I say they? Even I, if I don't have my phone near me, I think I'm isolated, lost. Now that has become an addiction, but we need to control ourselves. I don't say don't use it, but use it with control. Use it for genuine purpose, good purpose, because it's a remarkable medium to communicate. Yes, but we need to make sure that we do not lose our focus in the process. Research is something that, to me, research and teaching is not just a job; it's a vocation. It's part of life, and we must devote ourselves completely in this. We must submerge, and then only it works out. Like even today, if I'm late in my lab, I feel uneasy because something drives me that, okay, I should be there looking at something, doing something. Even at home, I read many things. I keep on doing work because that's and I think that curiosity to learn something new is very critical. It's very important. And that's where one more thing that's happens is, which unfortunately not happening in today's education, historical aspects, how concepts develop. See, we take for granted the DNA double helix, but how did it come about? We don't want to know. But if you want to know that, then yes, we can begin to design our own experiment. That how slowly, by step, sometimes by accident, sometimes by design, sometimes by perseverance, we get to know the deeper things, and that's what becomes critical in making things develop in a better way.
So one thing which, like, I really am going to keep it in my mind as a researcher, you said that science is all about conviction and not compulsion. So it was really motivating. So this was all
Yeah,
about.
yeah. I mean, you
Sorry.
must have passion to do it. Yeah, you were saying something. I'm sorry.
Yeah. So, no, sir. You can continue.
No, I mean, I can only emphasize. You see, like you asked earlier also that, okay, some people come to do PhD, and some people come to do PhD and research. You see, there are two components. Now, the difference, if I can redefine it, PhD you're doing, okay, as you take a course and you give an examination, you're qualified. Whether you're learned or not, whether you have been competent or not, you're not passionate about what you have done. You forget what you do your PhD and go somewhere else, and that's it. Well, that's also not wrong. You can change your path. But if you're not learned while doing PhD, if you're not asked your own questions, see, then what is the difference between a technician and a researcher? We can have very qualified technician. We tell them do this, do this, do this, and they come out and give us the results. But they have not. They won't be researcher. They may be author in the paper, but they are not research because they are not asking their own question. They're not defining their own way how to do it, and that requires deep knowledge, understanding, planning, analysis, and passion to do something. That's the point I wanted to make.
So this was all about science. So beyond science, when you are not doing science, how do you unwind and recharge yourself? As in, what are the activities or habits that help you stay creative, resilient, and motivated throughout your career?
Well, you see, there have to be hobbies. I don't have any strong hobby as such. I enjoy photography. In good old days when we were doing black and white photography, I learned how to develop a film, how to make my own prints. prints were for research, as well as my other photography that I did were printed by myself. I mean, that's one part of hobby. Today, of course, I can't be a regular photographer. When I go out traveling is one more thing that we enjoy, visiting some places, particularly natural sceneries or nature, or some archaeological structures, old structures. That's what I enjoy in my learning. And so I take photography. That's one part. The other part is enjoying music, classical music. That's another thing. I don't play, I can't sing, but I can listen. Classical music, yes, I've been exposed to that kind of sound from my childhood. And till date, we continue to enjoy. Actually, my wife also does it, and so they organize, my wife and daughter, son who is a cardiac surgeon, they also enjoy. So we organize musical concerts at our home, invite young artists. In a close family gathering, we enjoy that in our own. And if there are programs, yes, we do go. So that these are things. And then, of course, the other thing that I enjoy is doing household chores. To me, that's equally important, yes. Doing things at home, not just remain busy with my own things, but doing other things, whatever needs to be done in the home, house. I enjoy doing that. Sometimes I say, do it yourself person. I want to repair things, I want to do things, so that unwinds. Of course, I don't think research had wounded me deeply. So the unwinding is, to me, I don't know what does that really mean, because I never felt that I'm saturated with this and I have to do something. Everything comes on in natural time and natural process.
So I think these are the questions that our team designed. So Nina, do we have like audience questions?
Yeah.
Yeah. So you will take it up.
So yes, I will take it. Okay, so sir, there is first question. What is the difference between simply doing research and truly thinking like a scientist?
Well, that's tricky. I mean, basically, research is, you see, if you analyze the word research, researching. Something is not that we find something which didn't exist. It existed. We wanted to know what it is and how it is and why it is and so on, the mechanism behind it. Now, research can be just somebody who is doing research, or somebody who's just trying to find out, says, found this and says, okay, I've done it. But that has not created a new idea, new compulsions, or new views, or new information. A researcher has to be much deeper. It's not just, as I was telling earlier, that getting a degree is one part, but becoming a researcher, becoming a scientist is another part. Scientists need not have, as the common cartoon of scientists says, with very weird person. They are normal person. But then, it's not that scientists are better than, let's say, poets or painters or sculptors or engineers or something. Each one is doing their own job in a good way. Each one is a creator. Even a painter is, an engineer is, even somebody who's sitting in office doing clerical job is also a creator. So it depends on what you enjoy, how you do it, and where you add up something of value, something new to knowledge. In research, in scientists, we want to understand the nature. We want to understand the what we call mysteries of nature, be it physical, be it chemical, be it biological, be it space or whatever it is. Each one comes in that way, be it mathematics. But then we want to get deeper into the secrets of nature. That's what a scientist or a researcher would do, in my perception.
Yes, it was a very wonderful explanation. We would move on to the next question. Which is the best way to get pure DNA, either gel electrophoresis or centrifugation?
Well, both are very or you want the DNA fractionated. See, each method has its own limitations and its own applications. See, you want even DNA can be obtained by spooling. You see, good old days, when they didn't have high-speed centrifugation, the genomic DNA was simply spooled out after precipitating ethanol. Just put a glass rod and turn it around, and DNA gets wrapped around the glass rod. That's what we call spooling. That was also a method. Then came centrifugation, that okay, you pellet it because it's denser than water, or then came electrophoresis, but electrophoresis is useful only when you want to separate different sizes of DNA, which can be done by density gradient, but then that's more cumbersome, more expensive. Gel electrophoresis becomes a very simple way to separate DNA molecules of different sizes. And these sizes can range up to kilobase, or now there have been techniques of what we call as pulse field gel electrophoresis, where you could separate a whole chromosome of DNA. That's how the yeast chromosomes were separated in 1980s. All the 16 chromosomes could be separated intact chromosomal DNA by pulse field gel electrophoresis, which couldn't have been done by centrifugation. So it depends on what your objective is, you use a method.
Okay. So moving on to next question. In India, there are lack of government job opportunity after PhD zoology. How should I be motivated?
Well, yes, I agree. Jobs are there, but somehow the gap between the vacancy and being filled up, unfortunately, it's wrong. But then you see, the point is you decide what kind of a job you want, where it is opening. Unfortunately, one thing that I would speak out is we think after doing PhD we should get a job only in a university or research institution. I don't think that's the best way to look at it. School education, children need good teachers. They need good mentors, good motivators. And remember, school age is where most impressionable age. And I can tell you in a country like Finland, Norway, the brightest school child's ambition will be to be a school teacher. Why? Because that's most prestigious and most satisfying. So yes, I know jobs are becoming limited. Unfortunately, private institutions are also not doing ethically. They're kind of fleecing the young people, not doing it. But I hope the regulations come out properly. I know Supreme Court asked that all private universities should be audited how they are spending their money, but unfortunately that has not yet happened. And government colleges, government universities, state universities, central universities somehow are not filling positions. That's wrong. We have been talking about it. We have been raising these issues at different fora. But then I hope things will change. But in any case, what I would say is do not depend on a job. You can be your own. Now today there are many startup ideas also coming up. Young people can think of, if they have great idea, they have funding available, small amount, and one can develop many things. Yeah, there are difficulties, I don't deny, but then one has to find out what one can do the best.
Okay. Now, sir, question is, now that we have CRISPR and gene editing technology, if we remove oncogenes from our body in the near future, can we eradicate cancer completely?
No, oncogenes are essential for our basic life itself. Oncogenes are not present only in cancer; they are present in our normal cells, and they decide how we develop. They become wrong is when they become uncontrolled, when they begin to express when they should not be expressing, or when they become silent when they shouldn't be silent. So basically, it's not the gene itself is problem. The problem is with its expression because of either a mutation or some other change that gene begins to express. So if we can handle that, get its expression regulated. If that mutation if it's because of mutation, and if we can correct that mutation, yes. So techniques are there, but the question comes is we need to do a lot of animal experimentation to standardize. And then when it comes to humans, we have other considerations also to try. We can't just do an experiment. Okay, let's see what happens. With animals we can, with plants we can, but not with humans. And so that's where we have to be very sure. And the other major problem that happens is how do you target your engineering process to happen only in certain cells. I think we have drugs against cancer, but the major problem is how do we target those drugs only to cancer cells? That's one issue that still remains difficult one. But once we can do it, it may not be very easy because so this is where one problem we need to understand is we think this cancer cell expresses a protein and so we can target it. But what we still do not know is that protein expresses some other cell types, some rare cell types at certain time of development, and you block it or modify it, that can have additional problems. So that's how things go on. We take step by step. But then important point is don't think of removing oncogenes. They're essential for our life. Proto-oncogene as well as oncogene, they are absolutely essential for, because basically they are regulating cell cycle. When and how long or how many times the cell will divide. That's what primarily they control. And when they go wrong, cell keeps on dividing, and that's what we call cancer. But of course, this is a very simplified statement. There are many things happening, but we just can't remove oncogenes. No, that will be wrong approach.
Okay, so there is next question. There are some institutions they don't give the opportunity to the talent. They use reference during the selection of candidate.
Yes, that's a fact of life. I don't know what I can say about it. It's absolutely wrong. But you see, we don't live in an ideal society. We live in a society with a lot of competition, a lot of struggle, and therefore all these things happen. But what we can do, I mean, at least I believe is I should try it that I don't do a wrong thing. Others may do it. But then if more and more people begin to think that way, I don't think these wrong things will be eradicated. Never. They will always persist, part of living system. But then we can minimize it. And then, of course, the measure why these things more come is because of our extremely high population, limited resources, and therefore high competition. If we can bring a good balance in that, maybe these things will be minimized. They will never be totally lost, but they will be minimized. And the talent will find the right justified place only if we can develop that kind of condition. We need competition, but not the kind of competition that we had that for one post we have 10,000 applications. Obviously, nobody can select the best person in that process. We need a better system.
Yes, sir. So moving on to next question. We have lack of job opportunity in India even after doing masters from best college. What are the career paths for research?
Well, see, that's what I've been saying. Research is only one path, not the only path. Mind it. That's everybody who has done master doesn't need to do PhD. Everybody who has done PhD doesn't need to be an academic. Understand this. These are only steps where one can diversify. So, what if you think you are good in teaching and good in research? Yes, try for it. But if you think that's not your cup of tea, you're not and deeply interested in teaching or as you said, there are some people who are interested in research but not in teaching. Some who enjoy teaching but not research. Somebody who has done a PhD but neither enjoy teaching nor research. They need not continue. That they can find something where they can find a better way to satisfy their own inner human feelings and livelihood. So it's not that just because you've done master, therefore you need a college job or a university job, no, or a PhD, no. That depends. And of course, competition is there. And I hope we get into a healthier competition and a more honest competition, not the kind of things that are happening now. But many good institutions still have an honest system to decide. Of course, the way they do, there can be faults, there can be problems, but then they're not completely unacademic or wrong ways. So it depends on how well you are. Just because you have done your master's from a very good college doesn't guarantee that you are equally good either. Let me be blunt about this. You see, even there were toppers whom UGC said each topper from a university will be given a scholarship. I interviewed several of them and I wondered how they could become topper. So just getting that rank doesn't guarantee, unfortunately. What ultimately needs is what really lies inside you. That's more important for you to realize, for others to realize.
Yes, sir, absolutely right. So, next question. As we got to know, some of the US companies or startups, not taking a particular name, they are providing genetic optimization or embryo screening for the parents while IVF to create the best embryo for their family. Is it actually appropriate for the nature that we are changing everything to make someone best out of the best?
No, that's wrong. You see, eugenics, the subject that started in late 19th century, 20th, okay, late 19th century, and people thought that yes, we know that genetic things can determine what we are. Heredity is important. People did talk about eugenics, but that's completely wrong. See, we can't have everybody equally bright in every subject. They cannot be. Then you are not human being. We are just clones. We are just robots who are created identical. No, I don't think I will. And then the more important is, how do we know? Do we know the entire genome, what it does not? We don't know. We still don't know really very little about even not the protein coding genes. We don't know every protein coding gene, when does it express what it does. Leave aside the non-coding RNA. So unless we know that, we just can't say, okay, this gene is good and this gene is bad. No, it's not. Let nature take its own course. Much of the human problems have come about because we think Homo sapiens can control nature. No. To me, it's philosophically wrong. We are part, yes, we may claim that we are nature's best creation, but I sometimes jokingly say, did anybody ask a donkey what does donkey think of a Homo sapiens? We don't know. Because in compared to donkey, we may not do many things. And so likewise, as humans, we think that this is good, this is bad. That's a very relative way. So to me, that kind of engineering that's being done, it depends on specific genetic disorder. We know that, okay, this is the fact, and this the damage and that can be repaired, that's fine. But then to select what we call best genotype, we may as well be Hitler. That's what it did. Do we want to be doing that? No, not the knowledge of genetics, certainly. So to me, that's unethical in some ways.
Okay, sir, Muralidharan Raghavan is asking you for an advice. For after BSc or MSc in genetics, which course can I opt for forensic sciences?
Well, I don't know about all courses, unfortunately. I'm sorry, but there are courses which talk about forensic discipline. They can be diploma, they can be institutes of forensic science, and there many universities have forensic sciences. Or one need not have a degree. One can go and work in one such institution, get training, and go about that. That there are many ways, but I'm sorry, I can't give any specific because I don't have that expertise to talk about a specific course in forensics. But
Yes,
then the knowledge of genetic, knowledge of genotype, knowledge of looking at the DNA sequence and so on can yes be very useful.
yes. So next question is, what to do when you hear that colleagues has forgot data but you don't have proof and you can't ask those peoples.
I'm sorry, I didn't get the question clearly.
Sir, question is, what to do when you hear that colleagues has forgot data but you don't have proof and you can't ask those peoples.
Well, that's a very difficult situation. If you know it force and you don't have evidence, well, you can just raise a question. But then you see when that is published, there are ways to find out whether the data is false or not. Actually, good journals now want many times raw data as well to see. Because you see, research ethics has become a very important question today to ask that did you bypass some data? Did you generate through AI? There are now good journals can test whether data was generated through AI or generated what way. And that's where you see important point is an ethical requirement that every institution should do is that every research student must maintain daily record. What you have done, what you observed, that should be written down, noted in your daily logbook. Because that's evidence to say that yes, what I'm claiming is what actually I have done. That is something that we are still not learning, and unfortunately, that's where we go wrong. But that we need to do that. We need to enforce that kind of discipline. That you see now, let me put hypothetical that with your own student, you become suspicious that the student has created some data wrongfully. How to test it? You can look at students' record book, daily record book. If there's no record there, you have absolute good evidence to show that this was not done. Even if the student had done it but is not recorded, legally that becomes a situation that the student didn't do it. So to avoid that situation, we need to keep that record, not only from our competitors, but for our own sake. See, 10 years later, down the lines, you have published today, gone ahead. 10 years later, somebody asks, what is experiment really done? And that's where the experimental records become useful. For example, let me give this instance of Rosalind Franklin and Watson and Crick DNA helix discovery. Rosalind Franklin did indeed write in a notebook, and before Watson and Crick had come out with the double helix model, the DNA may be anti-parallel double helix. But now, how do we know this? Because she had maintained that record. Every day she would write, and she would write her impression. But it's a different matter that because of various compulsions, she didn't come to limelight, which was wrong. But that's what, at least after your life, you get people get to recognize that situation, that what everybody claims may not be correct. So it's necessary that we maintain our daily record, what we have done, what the results were, what our interpretation is. You need to learn that. Or I know today everybody is used hooked onto Excel sheet or something of that kind, but even right there itself. But make a note of what you think, what your impressions are from the experiment that you have done, what you have done today. That becomes useful. If that's not done, then yes, anything can be challenged, not by your friend, but by anyone else. That can be challenged.
Yes, sir, absolutely. Sir, Kumarika Shikha would like to know the areas of livelihood for a PhD zoology person.
Well, livelihood, you see, this is something that I've been saying again and again. The PhD zoology or botany, these are, yes, each area has its own plus points. One can develop a startup, one can develop one's own practical life. One can, you see, I sometimes think that even after, let's say, I've done masters in zoology, can I not go back, not go back to my own society, my own culture, my own, let's say, village or area where I can help them with that knowledge or something. We see we need to give up this habit of having a secure government job. Government job cannot be available for everyone who is born in the country. Everyone who has got to the highest degree, no, it cannot be. We need to find out something else, and that one has to find out where you are good. You may be good, let's say, have good drawing. Develop that habit with your zoology knowledge. You can do that. You may be good in describing something. You're good in using knowledge of zoology and developing a small industry, maybe poultry or maybe there are many ways. So it's not that there are only certain ways that can be defined. Okay, these are the paths that one can follow. It's entirely up to your individual capacity and interest. But do not expect that everybody will get a government job or a university or a college job. No, that will not happen. We have to be more industrious. We must learn to work hard with our own hands. That is something that's missing.
I think we have answered most of the audience questions. So, sir, first of all, what a great start to Sunday. It was very
Thank you.
insightful and motivating, and it was really an honor to ask you all these questions, and you were so candid about it. So it was a great opportunity to interact with you, and all thanks for making space for your time in your timeline for us. So yeah, I would like to now end the session, but first of all, thanks to great team who made all the effort to get sir here. So thank to you, sir, as well, and thanks to the SciComm Synapse team, all the backend members. Yeah. Okay.
I mean, I would like to thank you all. the entire team to have thought of me to ask me questions, and to the audience who have been listening. Maybe some questions were not what you would wanted an answer, but as you said, I just try to give my candid answer as I look at it and as I've experienced it. But thanks again for this opportunity. I really enjoyed it. Thank you so much.
We enjoyed it a lot, sir. It was like a great opportunity to pick your brain and get to know how actually things are working and were working. So it was really motivating. And yeah, thank you, sir.
My best wishes to all. Thank you so much.
Thank you,
Bye.
sir. Thanks, sir. So before ending the today's